Well, one big thing is that viruses are so incredibly different that, you might think, oh, we solved it all with SARS-CoV-2, we solved bacteria. No, we haven't. Every virus seems to be, throw a curveball and require different types of treatment.
All right, welcome to another episode of the Bioinformatics Lab Podcast. I'm Kevin Libuit with Andrew Page. We're from Pathogen, and today we're going to be speaking about M-Pox and the resources to analyze these types of genomes. Andrew, there's a ton of resources of which we've made available as well for analyzing viral genomes, especially when it comes into the context of Amplicon sequencing protocols, building a consensus assembly, and then going into downstream characterization. And that's been the blueprint, especially after SARS-CoV-2, with this really amazing, rapid wet lab approach to targeting a genome or a pathogen of interest, sequencing it, getting it assembled, and then getting it characterized in the appropriate mechanism. In MPOX, there's been obviously a lot of conversation about this with the current outbreak of the Clade 1b across the African continent, recently declared a public health emergency by the WHO. So what has your experience been in these resources to appropriately analyze Mpox genomes?
Well, one big thing is that viruses are so incredibly different that, you might think, oh, we solved it all with SARS-CoV-2, we solved bacteria. No, we haven't. Every virus seems to be, throw a curveball and require different types of treatment. So if you remember, like SARS-CoV-2 is like about 30 kilobases, Mpox on the other hand is like 200. And so, it's an order of magnitude bigger, and it's, obviously it can't be an RNA virus at that stage. It's a DNA virus, a double stranded. So immediately you got very different things. So it's either a very small bacteria or a very large virus. And it's complicated, right? And so people then try and apply tools that are built for one or the other, so for bacteria or for little viruses, and then they find they don't work. And so that's been a learning curve for many people, trying to straddle that, come from each direction. And with Mpox, it's quite interesting, and it's great that people immediately said sequencing is what we should do, and lots of sequencing has been done on it. And luckily, a lot of it has been going into the NCBI and open archives rather than proprietary archives like GISAID and locked away. Although they do have a kind of a build for it, and they pull data in from the public archives. But luckily, it's not being locked away with restrictive conditions. But Mpox sequencing is that a little bit more harder than SARS-CoV-2. And then analysis as well. We need slightly different ways of analyzing that data because, again, you're not dealing with a small virus, you're dealing with a large virus. And obviously, you know, doing phylogenetic analysis is that bit more difficult because you do have so much more ability to variation in there naturally. Um, so it's different. That's the takeaway.
Yeah. It's the same, but it's different. That's a really good frame to look at this because sometimes you can almost overemphasize the robustness of some of these approaches, especially when you're looking at amplicon sequencing to consensus to characterization. Even on the amplicon side, that this is always we're chasing the dog's tail, even with SARS-CoV-2. The just vocabulary of amplicon dropout is at the top of everyone's mind when they're looking at these types of investigations. And the same is true whenever you're looking at MPOX. We worked really closely with the Gruball lab in that rapid response, I think in 2022 or 2023 during that outbreak, on ensuring that there were primer schemes available and that we had the bioinformatics capabilities to analyze those genomes. But even then, on the rapid development and deployment, there was a conversation of dropout depending on even just the variants you were seeing in different regions of the world. So being able to have those readily updated amplicons, or rather primer schemes to generate the necessary amplicons for analysis is crucial and things that people should note along the way. And then from there, I would say the consensus assembly approach is more or less the same, but the biggest point of that conversation is the reference genome, obviously. So being aware of what you're actually aligning your read data to before you go into characterization. And now there are a ton of tools because generally the bioinformatics approach is the same with some just key variables you got to modify. Again, on our end, we've got the Theacove workflow that is built for this kind of analysis exactly, with a specific organism track for M-Pox. But of course, there's other resources out there. On our end, what we try to do is really just capture the best practices and make sure it's accessible to the laboratories we work with. But there's, we align ourselves with a lot of other international efforts. I know that Broad has repositories for both the metagenomic side and Amplicon specific side for characterizing these types of genomes, and even the CZ GenEpi resource. I think this is something you're familiar with, even with a recent update for M-Pox analysis.
Yeah, so they had to update their systems because obviously with the new outbreak, it's a different variant circulating, and it's gone through those so many generations now that it is different enough to the original outbreak a few years ago that actually they need to have a different reference, and so they've incorporated that into CZ GenEpi, and obviously that allows you to then do placement on top of a next strain tree, so you can actually see the context of where your things are, which is pretty awesome. But the original strains of Mpox haven't gone away. So say again, I was in Colombia recently, and they had about 100 per year of the original just kind of circulating, bumming around, and not a very major thing. It's just it's there, and it pops up every now and again. And obviously this new strain is doing the rounds now in Africa, and it's sort of bubbled over in a few places. But I guess luckily, for Mpox, it's skin to skin contact rather than airborne, so it does move at a much slower rate. And obviously skin to skin means that it is limited to more to certain activities and certain risk groups. I know last time in the UK, like, it burnt out pretty quick in the gay community, or men who have sex with men, and that is because there was widespread emergency vaccination, and then there was widespread alertness about what it is and how how to look out for it, and also then some people got infected as well. So basically, the availability of hosts diminished very rapidly, and then it just kind of burnt out. So whilst we thought it would be a huge thing, it turned out to be, a blip.
The beauty of the public health response in being able to communicate to the group at-risk populations, vaccinate even in this case as you're speaking about, and then ensure that it didn't spread in the way that it potentially could. And I think with this current outbreak with the clade 2B, or 1B rather, it seems to be a bit more infectious and a bit more transmittable. So that's why there's the concerns and all the efforts being put in play to make sure that that the frontline healthcare workers also can communicate this and have the resources to appropriately, hopefully, contain this current outbreak. Because I think with clade 1, especially across Africa and potentially other places around the world, it's more or less endemic. And as you said, it kind of just continues on and circulating through the population a bit. So being able to make sure that you have the resources to appropriately understand the spread of the clade 1 conversation is there, but in the face of this new sort of branching point and this new strain, or clade rather, of Mpox, you got to update the tools accordingly to make sure that you have the resources to appropriately watch what's happening in this continuously global network we have. So what happens in Africa, it's what happens everywhere.
I guess people are at the back of their mind, always a little bit cautious when it comes to any pox virus, because you had smallpox, and that caused absolute havoc for hundreds of years, throughout the world, and obviously it's really bad, and it's really great success that it was eradicated through vaccination. And what we don't want is something like that coming around again, and so people are hyper aware that this is something to keep an eye on, and hopefully this will just be something that bubbles up and then kind of disappears, and we'll just keep a careful eye on it. Fingers crossed nothing happens. But Genomics, luckily, is at the forefront of all of this, and most samples are certainly coming into middle or high income countries are being sequenced, and there is other surveillance efforts going on, to keep an eye on everything.
Yeah, that's right. So with that, thanks for another great conversation here. We'll be keeping our eyes on all the M-Pox resources. I know Phage is putting a couple different resources for folks to make sure that they are aware of the different bioinformatics solutions to analyze these types of genomes. So with that, we'll see everyone in the next one.